Hi, today we will discuss the different methods of identification of a person, such as the forensic anthropology, anthropometry, portrait parley, forensic serology, DNA fingerprinting, and forensic odontology. But first, what is identification? Identification is the determination of the individuality of the person or things. So now, let's go to the first method of identification, the forensic anthropology.
Forensic anthropology is simply described as the analysis of human remains for the medico-legal purpose of establishing identity. Forensic anthropology became established in the 1940s when the law enforcement agencies began to call upon physical anthropologists to assist with their cases that required identification of skeletal remains. So there are five main bones where the forensic anthropologists mostly interested in: the skull, humerus, pelvis, femur, and the tibia.
If you are thinking, what can we identify with these bones?
When it comes to a determination of sex, pelvis and skull are the best bone to be examined. When determination of race, the skull, and to determine the approximate age, the growth of long bones, such as the humerus, femur, and tibia. And also, the approximate stature, you can also examine the length of the long bones. For determination of sex, pelvis is the best bone to identify. Female have wider subpubic angle, and the sciatic notch of female is greater than 90 degrees, and female have a broad pelvic inlet.
That's it for pelvis. Let's go to skull.
The crest and the ridges in male are more pronounced than women. Chin significantly more square in males, and the forehead slopes more in males. Let's go to determination of race.
In determination of race, there are three race models to categorize skeletal traits.
First is the Caucasian, or the European, the Asian, and the African. Race determination is viewed as the critical part of the overall identification of an individual's remain. For the determination of race, for Caucasian, the nasal index is less than 48. The nasal spine is prominent spine, nasal ceiling, guttering, sharp ridges, prognatism straight, shape of orbital openings rounded, somewhat square. The determination of race of Asian, the nasal index is 48 to 53. The nasal spine, somewhat prominent spine, nasal ceiling, guttering, rounded ridge. Prognatism, variable, shape of orbital openings, rounded, somewhat circular. And for the determination of race of African, the nasal index is greater than 53. The nasal spine, very small spine. Nasal ceiling, guttering, no ridge. Prognatism, prognatic. The shape of orbital openings are rectangular or square. And now, for the determination of age from bones. Ages 0 to 5, teeth are the best bones to identify in this age because baby teeth are lost and adult teeth erupt in predictable patterns. And for ages 6 to 25, epiphyseal fusion, the fusion of bone ends to bone shaft. Epiphyseal fusion varies with sex and is typically complete by age 25. At the ages of 25 to 40, the bones is very hard. And for the ages of 40 above, basically wear and tear on bones, periodontal disease, arthritis, breakdown of pelvis, etc.
That is forensic anthropology. The next method of identification is anthropometry.
Anthropometry is the science dealing with the measurement of the human body, especially the head, body, and limbs, to determine differences in individual. It is introduced by Alphonse Bertillon in 1900s and spread it to the world. In 1914, fingerprint was officially adopted in France, replacing the Bertillon system.
Third method of identification is the portrait parley.
Portrait parley is a French word means speaking likeness. Portrait parley can be defined as a rigorous system for verbal description of physical characteristic of the subject. A distinctive description of every features of the face and head are especially valuable for investigation.
Especially when a set of the suspect fingerprint is not available in his file,
The fourth method
is the forensic serology.
Forensic serology is a scientific analysis of blood and other bodily fluids as evidence during the conduct of criminal investigation.
Before knowing anything, what is blood made of?
Blood is a mixture of many components such as cells, enzymes, proteins, inorganic substance, and water. But basically, the solid portion of the blood is composed of 44% red blood cells, 1% white blood cells, platelets, and 55% plasma.
In characterization of bloodstain, there are three questions that must answered by the forensic investigator.
First, is it the blood? There are presumptive tests that may use to determine if the stain is really a blood. The Kastle-Meyer, leukomalachite green, and luminol. In Kastle-Meyer, a solution of phenolphthalein is applied to suspected blood stain, followed by hydrogen peroxide. If there is a bright pink color is produced, therefore it is a blood. Next is leukomalachite green. It is the same procedure as the Kastle-Meyer, but it produces blue-green color if it is a blood. What if there is no visual of blood in a suspected area? Luminol test is used. If blood is present, it will glow. It must be viewed in darkness and photographed quickly. If the suspected blood stain confirm a blood, the second question is, it is a human blood? There is a test called precipitin test. It identifies the presence of proteins that are found only in human blood. For the precipitin test procedure, an animal, or usually a rat or rabbit, is injected with human blood, and the animal's blood form antibodies, and that antibodies are harvested from animal's blood serum, which called antiserum, and in the test tube, an extract from the suspected blood stain is added to the antiserum. If a precipitate forms where the two meet, then it is a human blood.
The fifth method of identification is the DNA fingerprinting.
DNA fingerprinting is a way to identify a certain individual rather than simply identifying a species or a particular trait. It is also a technique used by the scientists to distinguish between individuals of the same species using only samples of their DNA.
DNA fingerprinting was first reported by the English geneticist Alec Jeffreys. Where can we get DNA samples?
DNA samples can be obtained from blood, hair, saliva, semen, and other body tissue. The steps for DNA fingerprinting. First, you need to isolate the DNA. DNA must be recovered from cells or tissues. Only a small amount of sample is needed to isolate DNA. Step 2. The digestion of DNA by restriction endonucleases. The DNA is cut into fragments using restriction enzymes. Restriction enzymes or restriction endonucleases is a protein produced by bacteria that cuts the DNA at specific sites along the molecules, and the fragments cut by the restriction enzymes are called restriction fragment. Step 3. Electrophoretic separation of different fragments using the process called gel electrophoresis. DNA is negatively charged and it is attracted to the positive end of the gel. DNA is separated on basis of size. The shorter DNA fragments tend to move faster than the longer fragment. Step 4. Transfer DNA on nylon or nitrocellulose membrane. DNA fragments are transferred to a nylon sheet and placing it on the gel and soak it overnight by the process Southern blot. Step 5. Probe labeling. Adding radioactive or colored probes to the nylon sheet which is complementary targets to sequences. Step 6. Hybridization. The labeled probe DNA should be hybridized with a complementary sequence located on nylon membrane for detection of position of later. Membrane is washed to remove non-specific binding and clearing of the background. Step 7. Autoradiography technique involves alignment of hybridized membrane with X-ray film. The X-ray film, after its development, shows multiple number of bands that looks like barcodes and known as DNA fingerprints. And the last step, step 8, interpretation of band patterns, where the comparison of position of bands, also there and computer software that is available for the analysis of DNA fingerprints.
And last but not the least, the sixth method of identification of a person is forensic odontology.
Forensic odontology is the proper handling, examination, and evaluation of dental evidence, which will be presented in the interest of justice. Dental identification plays an important role when identification of remains of deceased person is skeletonized, decomposed, burnt, or dismembered, and is invalid by visual or fingerprint method. The teeth act as major source of DNA because of its ability to withstand the undergo changes. Some authors suggest that teeth are better sources of DNA than skeleton bones because DNA is found in vascular pulp, odontoblastic process, accessory canals, and cellular cementum.
That is the 6 scientific methods of identifying a person. Hope you learned something about our topic today. Thank you and have a nice day.